All right, well, thanks very much for the introduction. It's been a long time since I given a talk, and this is the first kind of live online seminar I'm giving, so I hope everything goes okay. All right, so I'm going to talk about a bit of the work we do in the plant-parasite interactions lab. And really the idea of the lab is kind of simple. We just want to understand enough about the fundamental biology of plant-parasite interactions from either the plant side or the parasite side in order to identify new avenues for control. And I'll give you an example of that from the nematode side today. So the choice of title for today is Sex, Organs and Food Security, the curious case of plant parasitic nematodes. So how can I justify that title, and what is a plant parasitic nematode? So this is my favorite picture of a plant parasitic nematode. Here you can see the nematode on the side of a root going from top to bottom. So what happens in this interaction is the nematode is born as a sexless juvenile. It'll then invade a root and move to vascular cinder, and then there it stops. And it chooses a single cell, and it will cause that single cell within the vascular cylinder to redifferentiate into this metabolically active syncytial feeding site, which sort of swells up inside the root. And you can see that highlighted, stained here in orange. These nematodes are biotrophs, so they feed from this tissue every sort of couple of hours for a period of several weeks in a non-destructive manner. And what I find remarkable about this is that the fate of the nematode and the feeding site are absolutely linked. So if at any time during this interaction the feeding site dies, then the nematode dies. And the reverse is true. And so if the nematode dies, then the feeding site will wither away and die. And so what that tells you is that these two are sort of actively maintained during this process. The second thing that I find remarkable about this is that this nematode sex is determined during parasitism by food availability. So if it makes a good feeding site and gets lots of nutrition, then it becomes a female. And if it makes a bad feeding site and gets poor nutrition, then it will terminally develop into a male. And so I think that's fascinating, but it's also important. So this kind of nematode, these sedentary endoparasites, have a worldwide distribution. There's at least one species for every major food crop of the world, including all of the ones here and many, many more. And together that costs well over $100 billion a year globally. Now that's a big number, but a combined cost of both the direct losses from the pathogen on the plant, so how it drains the plant resources and impacts yield, and also the cost of trying to control them with various inputs and practices. So what we're interested in is trying to understand this interkingdom communication, to understand what is it about the nematode or the plant that allows this prolonged biotrophy to function. And so when I first got into this area, we didn't know too much about this, although we did have two very important pieces of information. One was that the nematode delivers a bunch of proteins into and around this plant cell during infection, and we call those proteins effectors. And two, those proteins are compartmentalized in the nematode prior to injection into the host. So the nematode has these dedicated effector producing tissues or gland cells, and you can see on this schematic of sort of the business end of a nematode two sets of gland cells, the dorsal gland and the two subventral glands. So we knew of when I sort of got into this area sort of a handful of proteins, genes that are specifically expressed in this gland cell, and those proteins are delivered into the plant through this needle-like stylet at the tip of the nematode. And you may have seen that in the video on the right of this nematode, so pumping these proteins into the plant. Now, we didn't really know what most of these proteins were, but we knew that the nematode must know. So it must have some way of knowing, you know, in my genome, these are the genes that I want to express in the gland cell and deliver into the plant, or indeed, these are the genes that I don't. And so we reasoned that if we could find that regulatory mechanism, so how is it that a nematode knows, then in so doing, we could find all of the effectors and potentially also an attractive means to control the nematode. So what I mean by that when I say, you know, how does the nematode know? We're talking about tissue-specific expression, and so basically we need, you know, we're talking about promoters, transcription factors, and things like that. So to cut a long story short, we sequenced the genome of this animal. We identified all of the genes we knew were expressed in this tissue, and we identified their promoters in the genome. And then we simply compared those promoters to genes we knew were not expressed in the gland cell. And in so doing, we identified this motif, ATG-CCA, that's a non-coding DNA motif present in the promoter regions of genes expressed in this tissue. Now, this motif had two important characteristics. It was descriptive and predictive. So multiple occurrences of this motif in the promoter of genes described almost all of the genes we already knew were expressed in the gland cell and thereby delivered into the plant. And crucially, new genes, so genes that we did not know were effectors, but that had lots of copies of this promoter motif in their promoter, we could sort of validate those predictions by in situ hybridization. So we had to name this motif, and as usual in biology, when you're naming things, or often you start with your charismatic animal and then you sort of take steps backwards until you get to an anagram that you can sort of stomach. So we ended up with the dog box or the dorsal gland box. Now the dog box has two kind of major implications. The first one is that it basically showed us a more comprehensive repertoire of what are the genes that the nematode uses to manipulate the plant, and that was useful. But the second implication is perhaps more important, and that was that it implied the existence of a reader. So the fact that we had one non-coding DNA motif, and really only one, that unified literally hundreds, so over 200 genes specifically expressed in the same tissue, and these genes are otherwise sequence unrelated, implies the existence of some kind of reader. So some kind of protein or protein complex that can recognize this non-coding DNA motif and in so doing sort of orchestrate the expression of the effectors in that tissue. And so conceptually, we have this idea of all of the effectors at the bottom, and they're linked by this spatial regulator. Now, the best analogy I can think of for this kind of spatial regulator is like a conductor from an orchestra, because the conductor itself doesn't make any sound, but they're responsible for this sort of concerted action of a number of independent parts within this complex system. And we argue that if you had to remove only one person from an orchestra to make the biggest effect on disrupting it, then the conductor would be a good one to start with. And by the same token, of course, if you remove the nematode's ability to regulate spatial expression in this tissue, then that should be at least as good as the combined removal of all of the effectors individually. So it looks like an attractive target for nematode control and thereby crop protection. So we wanted to try to find this gene, go after this regulator. And so what I mean by that is we want to know which nematode proteins, and specifically transcription factors, although not necessarily transcription factors, could bind to this piece of DNA, this promoter motif. Now, there's many ways you can do that, but we had a nematode yeast-1 hybrid library in the lab. So we took one of these dog effector promoters, or rather a portion of a dog effector promoter that happened to contain 3 dog boxes within 60 base pairs. We screened it versus the library, and to our surprise and delight, all of the clones we got back were the same thing, and that thing was the DNA binding domain of a transcription factor. So that looked pretty good. We then cloned the full-length transcription factor, and the bottom panel shows you that pairwise interaction between the transcription factor and the dog box. And you can see that when you have both transcription factor and dog box, you get yeast growth evident of an interaction. And when we mutate the dog box, but we keep the bases the same composition, but only mutate the dog box, so leave most of the promoter intact, then we can abolish that interaction in yeast. So this looks like a pretty good candidate for this reader. We wanted to confirm that in at least one other way. So we purified some protein of this reader, crudely purified some protein of this reader, and then wanted to test it in vitro in an assay called an EMSA, or electron mobility shift assay. So the way this assay works is you take your same piece of DNA that you had before, you have it synthesized with a fluorescent molecule on the end, and then if you run that on a gel, then it will just sort of go as far as it goes. So it goes down to here. If you mix with that DNA some of your transcription factor of interest, if they bind, the complex is larger than the DNA alone, and so it migrates more slowly, and so the band sort of appears up here. If you then also mix in with that 100-fold excess of the cold dog box, so the same piece of DNA but without the fluorescent molecule, it outcompetes the fluorescent molecule, and so pushes it back down, and that you can see here. So we've used this assay both to confirm the interaction, as you can see, but also to confirm the specificity of the interaction by doing these kind of competition assays either with an unlabeled dog box or an unlabeled mutant dog box. And of course, you need much more unlabeled mutant dog box in order to outcompete the labeled dog box than you do unlabeled dog box. Okay, so that looks great. And then the last step, more recently, in collaboration with Thomas Baum and Thomas Meyer from Iowa State, we've been working on actually extracting the gland cells themselves from a nematode. So you can see that shown here in kind of blue and getting sucked into this needle, and then sequencing the mRNA of those gland cells. And we've done that for a related species, and the top most highly abundant transcription factor in this gland cell library is this transcription factor. It's nearly the top of any gene. I think it's the 22nd of all genes, so it's 99.97th percentile of all genes. And it's by far the most highly enriched transcription factor in the list. So we think that's even stronger evidence. So we're very bullish on this candidate reader now. We really think this is the one that binds the dog box. And so we're doing everything in our power to knock it out, either by classical reverse genetics, so sort of RNAi, or chemical genetics. And be happy to talk about those later. So we think we know what's going to happen when we knock this out, of course. The hypothesis is that when we knock it out, we'll knock out all of the effectors. And so we think that will be a very attractive way to sort of disrupt this nematode's ability to cause disease in plants. In parallel, we don't see any reason why this isn't going to work in other systems. And so it was a while ago that we found this motif in the potato cyst nematode, and as I told you, we called it the dog box. Around 2016, Maggie came to work with me while I was up in Scotland and with John Jones, and she wanted to work on pine-knot nematodes, and she wanted to basically do the same thing. She wanted to find a motif that described a non-coding DNA motif that described gland cell expression to help her find effectors in her favorite nematode. And to cut a long story short, she was able to identify a conceptually similar but sequence totally unrelated to the dog box motif in the pine-knot nematodes. So we had to name this one as well. In this case, we didn't use the charismatic animal algorithm. Instead, the motif itself is actually quite a cool name. So those of you who can read in DNA ambiguity code will know that a G or a C can be written as an S, and a T or an A can be written as a W, and a G or an A can be written as an R, and a G or a C and S. And so if you just read this in DNA ambiguity code, then it reads Star Wars, which we thought was cooler than any animal we could come up with. So we stuck with that for that motif. Now Maggie is starting a fellowship in Portugal to try to identify the corresponding reader of the Tata Wars in pine-knot nematodes. Now this discovery was really important for us because it said, yes, this is possible to apply the same kind of logic in other systems, even when they have independent origins of parasitism. And so therefore we thought, okay, if this is going to work in other places, where would be the best place? And so I turned to the root knot nematodes, which if you're familiar with plant-parasitic nematodes, you will know they are the #1 contributors to the crop losses due to nematodes. And they alone can infect, I think, all of the plants in this picture and many, many more. And so if there's going to be any impact of going after these readers and regulators to deliver crop protection, then I think the root knot nematodes is where it's going to be. Now, I can't share the sequence of the motif that we've identified because it's still in prep, but believe me when I say we have identified a conceptually identical to the dog box, but sequence unrelated motif in the root knot nematodes, and it seems to work in all root knot nematodes that we look at. We haven't named it yet. You'll be pleased to hear. And Caroline, rather unfortunately, joined the lab during lockdown in April on a Marie Curie fellowship to try to find the cognate reader of this motif as well. Okay, so that sums up the first half of this talk, and I wanted to use this example because it really, I think it's a good example of how, of what we try and do in the lab. Try to go from this fundamental understanding of some biological process in plant-parasite interactions and bring that to find novel routes to control. The second thing I want to talk about today, the only reason we're working on this is because it's interesting. And so this is not looking at the regulation of effectors, but actually looking at an individual effector family itself. Now, this particular effector family are called hypes, HYP, hype effectors. And the two least interesting things about hype effectors are that they are delivered into the plant. And so we know that because we have an antibody against one of these hype effectors, shown here in green, and it accumulates in the interkingdom interface, so between nematode and plant cell wall, shown in blue. And two, that they're important. So if we knock down hype expression using RNA interference, then we severely disrupt this nematode's ability to parasitize a plant. By far, the most interesting thing about hypes, though, is their genetics. Now, this started a long time ago. This started during my PhD, so before 2014. And we had just sequenced the first genome of a cis nematode, short read assembly. We also had life stage specific transcriptome information. And I was kind of, presented with this and could basically look at, my genes of interest. And so I set myself some criteria. I said, okay, I'm interested in biotrophy. So I'm going to look at only genes that are expressed in the biotrophic phases. I said bigger is better. So I only want to look at the most highly upregulated gene or genes in biotrophy. And I thought, PhDs are important. I want to do something new. And so I only want to focus on genes that nobody else has seen before. So I didn't want it to have, a similar sequence in this or be an ortholog of that. So if it had no blast hits whatsoever, then it was on my list. And so I went through that for the whole genome. I found a number of candidates, and you can see them here. And the top two, highlighted in orange, the ones that were subsequently named the hypereffectors. Now, in this short read assembly of the genome, there were two hype effectors, and they were quite similar to one another, and in fact, so similar that a single primer pair would amplify them both. So I designed a primer pair to amplify them both, and I get something like this. So you get this all the time, right? A major band, lots of minor bands. You clone your major band, it is what you expect, and you kind of forget about everything else. Well, I was expecting two. We cloned the major band and we sent like 10 for sequencing, and then I think like 7 or 8 came back different. And not like a little bit different, like very different, but still clearly the same gene family. And so I thought, okay, sent another 10 and another 7 or 8 came back different, and this went on for some time. And in the end, we sort of accumulated 75 unique genomic clones that could all be grouped into this single gene family. And really, at that point, it's not exhaustive. We just stopped because we weren't getting anywhere, but we could have just kept going for who knows how long. All of these sequences could be grouped into one of 3 different subfamilies, subfamily 1, 2, or 3. What those subfamilies shared in common was this highly conserved region at the beginning and the end of the gene. And it's 95% conserved within subfamily or 99% conserved between subfamily. How they differ was this, what we later called a hypervariable domain, which was in the middle of the middle exon. And this hypervariable domain contained subfamily-specific, variable number, variable organization, and variable sequence repeats. Now, I steer clear of the word tandem repeat because that sort of, to me at least, implies lots of different numbers of copies of the same thing. And actually, most of the variability in this hypervariable domain is variable organization of repeats that themselves are sequence variable. So to try to explain what I mean with a picture, for subfamily 1, within this domain, individual genes can have anywhere between 3 and 4 of these motifs, and you always have motif 1.1 and motif 1.2. The positions of these and the numbers of them vary dramatically from something like, I think this is not up to date, I think the minimum is something like 4, and the maximum is something like 27, and the organization of them varies tremendously. Now, I'll come back to that at the end. But what really struck me and still strikes me as crazy every single time I think about it is that all of the variants we've ever found are in frame, and this is not splicing, these are genomic variants, these are genomic clones, and this is within the middle exon, and the middle portion of the middle exon. And every time we have a novel organization, they are always in frame with the remainder of that exon, and I'll come back to that. So when we found this out and we found this sort of large gene family, we sort of thought, well, how does this work? We know nematodes are small, so when you're sort of cloning genes from nematodes, you don't clone from one nematode, it's too small. So you have thousands in your prep, and then you extract DNA and clone your genes. And so we thought, okay, what are the possible scenarios? So is it true that every nematode has all of these? Is it true that each nematode has just one? And the reason we see this big gene family is because we sequence thousands of individuals, or is it sort of somewhere in between? Now, obviously, to get at this, we have to look at individual animal basis. And so if we take a single female, we extract DNA and we look at the hype complement for either hype 1, 2, or 3. To our surprise, no 2 individuals had the same genetic complement of hype effectors. All nematodes had at least one, but none of them were the same, and they varied dramatically. So for example, 2 nematodes, same population, same plant, same part of the root, one of them could have just 1 copy, and the next one could have 20 or more copies. And not like 20 copies of the same gene, but 20 different versions that have been shuffled only in that middle exon. So that doesn't happen, right? That's unusual. And so variation is between individuals of the same population, and therefore each nematode has some of these genes. Now that's really where we left it at the time, so 6 years ago, because we really couldn't go any further. The technology wasn't really sufficient that we could go any further. But since then, technology has advanced quite a lot. Sequencing technology has advanced quite a lot. And we started thinking about this again. And so we thought, okay, how is it possible that a genome can permit this level of variation? How can it be that you have one gene, I have 20, and yet they're shuffled in some way, and yet somehow we're still sort of compatible? And so we came up with some hypothetical scenarios. Scenario one for this diploid organism is that hype loci transpose, so they're like transposons. So maybe hypes jump around in the genome, and so the two haplotypes of an individual may differ from one another, but importantly, individuals would differ from one another in both their number and position of hype loci, and that would explain what we've seen. Or scenario two, and these aren't necessarily mutually exclusive, is that there is a hype locus, or there are, major hype loci, and you get this kind of local rearrangement and presumably tandem duplication, either on one haplotype or on both, although again importantly such that individual 1 is different from individual 2. And again, this would explain what we've seen. So in order to answer this question, obviously we need to sequence genomes of individuals, and technology hasn't advanced that far. That's still impossible. But the game changer was nanopore sequencing. So nanopore sequencing is different because it actually sequences DNA. So if you put a piece of DNA into a nanopore, that strand of DNA goes to the pore and it is read. It's not amplified or chopped up or anything like that. And so we reasoned that even if we sequence the genome of thousands of nematodes all at the same time, yes, we'll never be able to say these 2 reads came from this worm and these 5 from this one, but we will be able to say one read came from one individual for sure. And so in so doing, we could identify these sort of ultra long reads from individuals. We could find those reads that have a hype in them, shown here by the black boxes. And then we could use the non-hype genes on those reads, shown by the colored boxes, to align that read to the genome, like a consensus assembly of the genome, and thereby distinguish between scenario 1 and scenario 2. So we bought a nanopore, and Unati, who I think is signed in here, probably hates me at the moment for sharing this picture, but I really like it. This is the day the nanopore arrived, and this is happiness. So Unati sequenced the genome of these animals. We identified all the reads that contained hypes. And the first thing I should tell you is that no single read had more than one hype. And so immediately we looked at scenario 2 and we said that's just not possible. And then we mapped them to the genome, and this is what we saw. Actually 2 hype loci. So all of the hype 1 containing reads map here, and all of the hype 2 and 3 hype 2 and 3 containing reads map to a different locus. So we're scratching our heads, right? This doesn't make any sense. And if you zoom in on these, they really are single loci. It's a beautiful peak. And if you zoom in even further, this is the hype locus. So you have exon 1, exon 2, exon 3, and each individual bar, gray bar, is a separate read. The purple bits are indels, and obviously these are noisy reads. This is nanopore, but I hope you'll appreciate almost all the variants stack up in the middle part of the middle exon, that bit that gets shuffled. So the conclusion from this cannot be any other way other than that hype variation is allelic. Now this sounds like trivia, but this is really important because, think about what this means. So you're born as a single cell, and you're diploid, and so you can either be heterozygote or homozygote. And so at worst, you can have 2 versions of this allele. You can have 2 hypes. Although we know by the time you get to an adult, you can have many. And I sort of say, colloquially 20, but God knows how many. I mean, we've only looked at 6, so it's many. And so the only way to reconcile this, and still in an adult, it's a single locus. So the only way to reconcile this is that at some point during the lifespan of this animal, so in 14 days, it has created alleles at the same locus. So it has somehow shuffled the hypes that it has and generated more hypes at some point between cell divisions. Now, I hope I don't need to tell you that that's just totally unprecedented. Genes don't work like that, and you can't just create alleles well, not normally, you can't just create alleles so rapidly and in such a bizarre way. Now we know that the shuffling is highly constrained. It seems to be constrained to these loci. It seems to be constrained not just to the gene, but really the middle exon, the middle part of the middle exon of just the gene. Bizarrely, it seems to be constrained by frame, which boggles my mind because DNA doesn't know frame. It also seems to be constrained by cell division because it doesn't happen every cell division, because here you have 2,000 cells and here you only have one cell, and we don't get 2,000 variants. We're fairly sure of that per animal. So it's happening sometimes, but not all times. And also, there are a series of rules of engagement to this shuffling. It's definitely not random. You know, we calculated what would be the number of variants we would expect to see if it were random, and what do we actually see? And it's definitely not random. There are, you know, within that hypervariable domain, there are some motifs that sort of only occur at the beginning or often occur at the beginning. And there are some that often occur at the end. There are others where you never see it adjacent to itself. There are others where you see it adjacent to itself in stretches, but they never go beyond 6, let's say. And so there are definitely some rules of engagement to how this shuffling is going on. And I just think finding that out is going to be really fascinating. So we're trying to answer loads of simple questions. You know, how is this happening? What's actually doing the shuffling? Why is it happening? And when? And that's quite linked to why. The only thing I will say, the 2 annotations I'll add to this is the infecting of the plant happens here. So just after the juvenile, and everything up until that point is mitosis only. Meiosis, so these are born sexless, you know, juveniles, and as I said, their sex is determined once they're in the plant, and so meiosis happens later. So when does this happen and why? My gut feeling, although I could be wrong, probably wrong on this one, my gut feeling is it's happening some point in the adult, or the majority of it's happening some point in the adult, simply because the adult knows when it's failing. So I told you that their sex is determined environmentally, and so, you know, if it's not going well, if parasitism is not going well, they become a male, and if it's going well, they become a female. So it has some mechanism of knowing parasitism is not going well. And so if parasitism is not going well, it might attribute that to its effectors. So these effectors are not okay. I want to shuffle them and pass them on to the next generation, the shuffled versions, but not randomly shuffle them. That would be crazy. I just want to shuffle them in some constrained way such that they might then work better in the next generation. I will say, though, Unati has already looked at the juvenile, and it's definitely started to happen already at this stage. And so that prediction, you know, that sort of hypothesis would predict that a juvenile does less shuffling than adults, males do more shuffling than females, and, you know, females differ in their fecundity, so the more fecund might do less shuffling than the rest, and so we're working through those. Okay, that's all I want to say on that. I'm very interested to hear comments and questions about this, and any ideas for sure. With that, I just want to thank everybody in the lab that does the work and makes it a joy doing the work. I have to thank collaborators and friends equally from all over the place, and in particular, I've got to thank funding bodies, BBSRC, that's sort of the major funder of the lab, but also Marie Curie, EMBO, and the Rank Prize Fund, Isaac Newton Trust, Wellcome Trust, and the Carnegie Trust. And thank you very much for your time, and I'll pass back to the chair, I think.
No, you can leave your slide up there, Seb.
Okay,
so
sure.
Thank you very much for showing such interesting and totally unpublished data as well. That's really exciting for everyone to hear, that it's really new stuff, and obviously stuff which is a little bit confusing at the moment, indeed. So yeah. Let me start by just asking, so you mentioned this is completely unique, so you've seen nothing like this in any other system that you're aware of? Initially I thought about Huntington's disease, and you get these, you know, triple repeats and things, but this is obviously a larger motif, I'm guessing.
Yeah, it's definitely unprecedented. I mean, there are lots of examples of, you know, gene families or clusters of genes that undergo some kind of somatic variation, or, for example, you know, rearrange and things like that, but never restricted to a single locus, and never restricted in the way that I sort of show here. So the something is definitely new. It can't be described by sort of known mechanisms of, you know, unequal crossover or anything like that.
I see. Okay, so I encourage everyone to ask any questions in the question box, and then I will relay them to Seb. So I have a question from Pavithran Narayana, and they ask, can the variation in hypes provide infection advantage or disadvantage to individual nematodes? I guess this might be challenging to do, but do you have an idea about that?
Yeah, we do. So I don't know. I sort of expect so in a sense. So my gut feeling is that it sort of has to in some way. It must be a reason for this sort of madness. I think we would be able to test it. So the idea is, we sort of reason that if the nematode effectors determine how good it's going to be as a plant parasite, and how good it is as a plant parasite determines if it's male or female, then, by following that logic on, it should well be that males are sort of unsuccessful, if you like, and would contain unsuccessful alleles, and females would contain the successful alleles. And so by looking at males and females, we should be able to see that their hype complements differ in their parental hypes, so the ones that they were born with. Yeah.
Right.
Yeah, great idea.
Yeah, so another question from the same person, and this is kind of maybe a more basic question about nematode biology, but they ask, do nematodes derive nutrition from the soil before infecting a plant, and do soil nutritional deficiencies affect the infection, both from the nematode and the plant's perspective?
Yeah, very interesting question. So the answer to the first one is no. So they don't. All of their external nutrition is derived from the plant throughout their entire life. So they're born as a juvenile, they don't feed until they get into the plant, and only once they start that biotrophy do they start feeding, and once they start feeding, they never stop. So they can't sort of go backwards and sort of make a different feeding site. That's it. So they just feed and then lay eggs in their own body if they're lucky and become a female. Whether the nutritional sort of makeup of the soil has an impact, I don't know for certain, but I would sort of assume yes, because it matters for the plant. So if the plant is not fit, then the nematode doesn't do well, and so you get more males. And so indirectly, I would say yes through the plant, although I don't know that for certain.
Okay, so I think you've blown most people's minds. The question, oh, here we go. There's another one. So William Rooney asks, do you know whether these hypervariable regions from the hype effector are translated? No.
Oh yes, I should have mentioned. Yeah, they are. So they're in the transcriptome. It's very difficult for us to, and we've cloned them from cDNA, et cetera, et cetera, et cetera. So yes, and they're always in frame. It's very difficult for us to know which transcript comes from which gene. One, just because of numbers. I mean, there are so many of these things, we don't have an exhaustive list of all of the genes, let alone all the transcripts, and so marrying them up is difficult. And two, we actually know that, unfortunately, reverse transcriptase introduces non-canonical jumps along the hype region. And so we've steered clear for now of looking at transcripts simply because we can't trust cDNA. Now that we have nanopore and we can actually sequence RNA without going to cDNA, so direct RNA-seq, we will do that. So we will look at that, and indeed, we've already done this for one species, and we get loads and loads of hypes in the transcript as well.
Okay, cool. Okay, so I think we'll go on, sorry. Go on.
Well, just the last point. I should mention we definitely know the translators as well. So if you remember, we have an antibody against these, and we can recognize them.
Yeah, that's true. Okay, so I think we'll leave it there. I'll remind everyone that we're gonna have a recording of this. We'll be online. So if it's blown your mind, rewatch it and then get back to Seb with any questions that you have. I have maybe a slightly flippant question just to end. So it struck me it was very brave of you as a PhD student to look for something which didn't blast to anything else. I'm not sure, is that a strategy you would recommend to people these days?
No, definitely not. It was stupid, I think. I mean, yeah, the downside is you've got nothing to go on. So like, for example, we have no idea what these genes do still.
Okay.
Yeah. So the interesting thing now we're looking at is their genetics, but we still don't know what they do. And so at least if we had some blast hits, we might have a handle on that. So I would definitely not recommend it.
Well, it's going to be a super interesting story when you piece it together for sure. So so thanks very much, Seb, for talking to us today. That's really great. Okay.